Imagine trying to follow, like, the plot of this complex, sprawling movie, but 80% of the scenes are completely missing the main actors.
Right, that would be impossible to watch.
Exactly. I mean, you have the script, you know all the locations, but you have no idea who is actually standing in front of the camera on any given day. And, when we usually talk about AI and genetics on this show, the focus is almost entirely on reading that script, just scanning an endless sequence of DNA, the A's, C's, T's, and G's.
Yeah, and to be fair, that has been the prevailing approach for, well, the last decade. Decoding the static architecture of the genome is just a monumental task in itself. But it only gives us the blueprint. It really doesn't tell us how the building is, actively managed day to day.
And that brings us to the sources we're looking at today for you, our listener. Because for this deep dive, we are pulling from a really groundbreaking 2026 paper published in Cell Genomics. It details a new genomic foundation model called ChromBERT.
It's a fascinating piece of work.
It really is.
Yeah.
So the mission for this deep dive is to completely unpack how this AI model does something entirely different. It's not just reading the DNA code, it is decoding the incredibly complex, unwritten language of the proteins that act on that DNA, the ones turning genes on and off.
And, crucially, it's solving a massive problem in biology where physical data is just frustratingly sparse.
Yeah.
And it's doing it without needing a single new physical lab experiment.
Okay, let's unpack this. If the DNA sequence is the script of that movie we mentioned, the transcriptional regulatory networks, the transcription regulators themselves are the directors, the lighting crew, and the actors.
That's a great way to think about it.
Because every single cell in your body has the exact same DNA script. But, a liver cell performs a completely different function than a neuron. The TRNs are the ones deciding which scenes actually get filmed in a specific cell to make it act like a liver cell instead of a brain cell.
Yeah, they are essentially the operational control systems of human biology. These transcription regulator proteins, they physically bind to specific regulatory regions of the DNA to either amplify the expression of a gene or just silence it completely.
Right.
But the constant headache for biologists isn't reading the DNA script anymore. It's figuring out exactly where those protein directors are standing on the genomic stage at any given millisecond.
Which is incredibly hard to do.
Oh, absolutely. To capture that, researchers rely on this experimental method called ChIP-seq. It's a highly specialized physical wet lab experiment that acts kind of like a camera, capturing a snapshot of exactly where a specific regulator is bound across the entire genome.
I want to pause on this for a second because I'm sure some of you listening are wondering, wait, hasn't the ENCODE project already mapped all this out for us? I mean, we've been hearing for years about these massive international efforts to map the human genome's regulatory elements.
Well, yes and no. I mean, ENCODE is a monumental baseline for sure, but it primarily covers highly researched, heavily studied cell lines. Here is the actual physical bottleneck. There are roughly 1,000 known transcription regulators in human biology.
1,000. Oh,
Right.
yeah.
So doing a dedicated ChIP-seq experiment for every single one of those 1,000 regulators, and then multiplying that by every single unique cell type in the human body.
Oh, wow. Yeah.
And then multiplying that again by every possible state of health, disease, and developmental stage.
Yeah.
It's just the combinatorial explosion makes it astronomically expensive. It's logistically impossible.
So we basically have the script, but we are missing the paparazzi photos of these protein actors for the vast majority of the human body. We only know what they're doing in the most famous, glamorous locations, right? For everything else, the data is just totally sparse.
Exactly. And without knowing where those regulators bind in those obscure or, say, diseased cell states, our understanding of how complex diseases develop or how cells evolve over time, it just hits a brick wall.
All right, so tracing our steps so far for a quick recap, we have these transcriptional regulatory networks directing the DNA script. We have ChIP-seq acting as our extremely limited camera. And we have this massive gap in our foundational knowledge because physical lab work simply cannot scale to map every cell type.
Right, that's the core problem.
So think about your own field of work for a second, right? How do you build an accurate, reliable, predictive model when 80% or more of the foundational data you need just doesn't exist? That is the exact puzzle ChromBERT was engineered to solve.
Exactly.
Since we can't physically test every cell, we need an AI to infer the missing pieces. So let's dig into the architecture. I mean, how do you even begin to pre-train an AI to understand the syntax of proteins?
Well, you have to start by feeding it the largest repository of physical data we currently possess. So the researchers compiled this data set called cistrome human 6K.
Okay, cistrome human 6K.
Yeah. A cistrome is basically the complete set of binding targets for a specific regulator across the entire genome. This dataset contains about 6,391 of these cistrome tracks. It pulls together data for about 1,000 regulators from over 100 different tissues and cell types.
But throwing all of that raw data into a model at once would be like computational chaos, wouldn't it? There is a very specific structural mechanism they use to feed this into the AI, right? I read that they chop the genome up into 1 kilobase intervals.
Yes, that's exactly right. They partition the entire human genome into over 2.1 million of these discrete 1 kilobase bins. And for every single bin, the AI evaluates the presence or absence of those 1,000 regulators.
So imagine a massive 1,000-lane highway, but we're only analyzing a stretch that is exactly 1 kilometer long at any given time. Each lane represents a specific protein regulator, right?
That's a great analogy, yeah.
So the AI is essentially looking at the traffic pattern, who is parked in which lane, and which proteins tend to cluster together in this specific 1 kilometer stretch.
Precisely. And by scanning over 2.1 million of these distinct stretches, the model begins to internalize the underlying mathematical rules of protein interactions. It does this using an 8-layer transformer encoder, which is the BERT part of ChromBERT.
Right, bidirectional encoder representations from transformers.
Exactly. And it uses a masked learning strategy.
Now, I know masked learning is pretty standard in large language models, hiding words in a sentence and forcing the AI to guess them based on context. But how does that apply to a 1,000-lane biological highway?
So during the pre-training phase, the algorithm randomly hides, or masks, about 15% of the binding statuses within a given bin. The AI's task is to analyze the unmasked 85%. So the spatial context of all the other regulators present in that specific stretch of DNA, and then predict the missing 15%.
So it is literally a biological game of fill in the blank. Like if the AI consistently sees protein A and protein B parked next to each other, it learns the grammatical rule that protein B usually follows protein A.
Right, it learns the syntax.
But wait, there has to be a major mathematical hurdle here. In any given stretch of the genome, the vast majority of those 1,000 lanes are going to be completely empty, right? Unbound statuses are way more common than actual protein binding events.
Oh, absolutely. That is the class imbalance problem. And it is severe in computational biology. I mean, if the AI simply optimized for raw accuracy, it could just guess unbound 100% of the time and be right, like 99% of the time.
Right, which would give it a great score, but make it totally useless.
Exactly. It renders the model completely useless. It's the classic needle in a haystack scenario. If you guess hay every time, you guarantee a high score, but you never actually find the needle.
So how does the architecture actually force the model to care about the needle?
The researchers implemented this mechanism called focal loss. Now, a standard machine learning loss function applies a uniform penalty across all errors, but focal loss dynamically reshapes that calculation.
How?
It introduces a modulating factor that exponentially reduces the penalty for making a mistake on the easy, highly probable majority class, which is the empty space. And simultaneously, it heavily penalizes the model for getting the hard, rare minority class wrong, the actual binding events.
Ah, I see.
Yeah, it fundamentally alters the gradient descent process, mechanically forcing the AI's attention weights to focus entirely on learning the complex grammar of when proteins actually bind, rather than just, coasting on the statistical base rate of empty space.
Okay, let's pause and synthesize this pre-training phase real quick, because the mechanics here are just brilliant. So, summary bullet points for you listening. They feed this massive cistrome 6K dataset into the model, chunks into these 1 kilobase bins.
Yep.
By hiding 15% of the data and forcing the model to guess who's missing, the AI learns the fundamental social network of these proteins. And it does this using focal loss, so it actually learns the complex grammar instead of just cheating the test by guessing empty every single time.
That's a perfect summary.
It really makes you wonder, what stands out to you here? For me, it's that if an AI can learn the underlying syntax of human molecules, what other hidden complex languages in nature are just waiting for a transformer model to decode them?
Oh, the possibilities are endless.
But I am struggling with a conceptual leap here. Pre-training gives the AI this broad, generalized knowledge of protein grammar. It knows how actors behave generally, but a liver cell's environment is totally different from a neuron's.
Right, very different.
So how does it predict what happens in a specific unseen cell type? I'm assuming you don't just type, please act like a liver cell into a text box.
No text boxes here. You're touching on the transition from pre-training to task-specific fine-tuning. Once ChromBERT internalizes that core protein grammar, the researchers freeze the early layers of the neural network.
So they lock in that foundational knowledge so it doesn't get overwritten.
Exactly.
Yeah.
However, they leave the later layers, which act as a lightweight decoder, unfrozen, so it can be rigorously trained for highly specific biological tasks. And to give it the context of a specific cell type, they use prompts.
Okay, prompts. But I'm really trying to picture what a prompt looks like mathematically in this context. If it isn't text, what is the AI actually ingesting?
To understand this, you have to look at what the frozen layers of ChromBERT are outputting. They output embeddings. And an embedding is basically a high-dimensional mathematical vector. In ChromBERT's case, it's a 768-dimensional space.
Wow, okay.
Yeah, it takes the state of a regulator and plots it as a coordinate in this incredibly complex mathematical space. So a biological prompt is just additional data that is mathematically concatenated, fused directly onto those 768-dimensional TRN embeddings before they pass through the decoder.
Okay, give me a tangible example of the data they're fusing. Like, what does that look like?
Well, a primary example is DNA-seq data. This is an experimental track that measures chromatin accessibility. Basically, it maps out how physically open or closed the folded DNA is in a specific cell type.
Right, because if it's closed, nothing can bind to it.
Exactly. If the DNA is tightly coiled, proteins cannot physically access it to bind. So by concatenating this accessibility data onto the embeddings, the prompt acts as a mathematical attention mask.
Ah, so it's like the AI already knows the general motivations and habits of the actors, but the prompt hands the AI the specific stage design for tonight's play.
That is exactly it.
It mathematically tells the model, hey, ignore this entire section of the stage, the curtain's closed there, just focus your predictions over here where the stage is open.
That is a perfect way to visualize it.
Yeah.
And they also utilize RNA-seq data, which measures the actual output of gene expression. They run that through another foundation model called SCGPT to generate prompt embeddings.
So they're stacking models.
Yes. And the results of providing this context are just staggering. Look at the cistrome imputation case study from the paper. This is the ultimate test, asking the model to predict unseen binding sites for a regulator in a cell type it has completely never encountered before.
The ultimate test of generalization. So how did it stack up?
Well, they benchmarked ChromBERT, using those DNA's prompts, against the previous state-of-the-art model in the field, which was called Avocado.
Avocado, okay.
And the performance metric here is crucial. It's the AUPRC, or area under the position recall curve. It's a rigid measure of how effectively a model identifies true positive binding events without just drowning the user in false positive noise.
So quality over just raw guessing.
Exactly. Now, Avocado was hovering around an AUPRC of 0.514, which is essentially slightly better than a coin toss when balancing precision and recall.
Ouch. And ChromBERT?
ChromBERT achieved a score of 0.659.
Wow. I mean, in the realm of computational biology, jumping from 50% to nearly 66% on precision recall curve, that isn't just an incremental software update.
No, it's not.
That is a paradigm shift in predictive power. So let's pull this fine-tuning section together. We take that generalized protein grammar, we freeze it in the early layers, and then we use biological prompts, like physically mapping out the open and closed areas of the DNA to give the AI specific stage directions.
Right.
And this allows it to predict missing data and find those missing paparazzi photos with an accuracy that leaves older models completely in the dust. I mean, think about your own decision-making process for a moment. If you wanted an AI to accurately predict your next career move, what hyper-specific contextual prompt from your daily life would you need to mathematically fuse to its base logic?
That's a great question to ponder.
It really is. But moving beyond the architecture, knowing where proteins bind is an incredible computational feat. But what does this actually mean for human biology and treating diseases in the real world?
This is where ChromBERT really transitions from a marvel of computer science into a highly practical medical tool. The paper highlights several applications, but a major one is EQTL fine mapping.
Okay, let's break that down. For those not steep in genetic pathology, EQTL stands for Expression Quantitative Trait Loci. Essentially, these are slight genetic variants, tiny mutations in your DNA sequence that directly alter how much a particular gene is expressed.
Yes.
And these are frequently the exact underlying culprits linked to major diseases, right?
Exactly. The clinical challenge is that when you find a genetic region associated with the disease, there are often dozens of variants just clumped together. You need to isolate the truly causal variant, the one actually disrupting the regulatory network, from the innocent bystanders that are just, genetically linked to
Well,
it.
finding the actual bad actor in a crowd.
Exactly. To solve this, the researchers fine-tuned ChromBERT using a DNA sequence prompt generated by another model, DMBERT22.
So they feed the DNA sequence data through one AI, turn it into a prompt, and fuse it to ChromBERT's protein network data to hunt for the actual culprit. What was the verdict?
ChromBERT achieved an ROS area under the receiver operating characteristic curve of 0.828.
Okay, give me some context for that number.
Sure. To put that into perspective, Informer, which is a highly respected sequence-only model, scored 0.770. Even the DNever-2 model operating independently only scored 0.788.
So ChromBERT is significantly more adept at identifying the precise disease-causing mutations.
Yes, precisely because it isn't just looking at the DNA typo. It fundamentally understands how that typo physically disrupts the protein interaction network above it.
Here's where it gets really interesting, though. Beyond just mapping diseases, the paper dives into cell state transitions and transdifferentiation.
Yes, this is fascinating.
Transdifferentiation sounds like cellular alchemy to me. It's the biological process of reprogramming one mature, fully formed cell type directly into another. Like taking a fibroblast, which is a structural connective tissue cell, and magically turning it into a macrophage, a vital immune cell.
It is basically cellular alchemy, yeah. But it's driven by very specific regulatory mechanics. To achieve that transformation, you have to know which specific transcription regulator to perturb. Which protein is the master key that forces the entire cellular network to rewrite its identity?
And historically, identifying that key driver required endless, expensive, trial-and-error physical lab experiments.
Yes, years of work. But ChromBERT can identify these key driver regulators virtually by calculating something called an embedding shift.
I really want to break down the math of an embedding shift because this is wild. We established earlier that ChromBERT translates every regulator's state into a 768-dimensional mathematical vector, right?
Right. So what the model does is it isolates the mathematical vector for a specific regulator when the cell is in its initial fibroblast state. It then calculates the vector for that exact same regulator in the target macrophage state.
Okay.
And then it measures the geometric distance between those 2 points in that 768-dimensional space.
Yeah.
If a regulator's vector has moved a massive distance, which is a significant embedding shift, the AI flags it.
Wow. So the math is essentially saying the interaction network and biological behavior of this specific protein radically transformed during the state change. Therefore, it is highly likely to be the primary driver of the transformation.
Exactly. It isn't just guessing. It's measuring the mathematical distance of social behavior changes among proteins to print out a ranked hit list of which proteins are driving the evolution.
That is incredible. But did they test this against reality?
Oh, they did. They validated it against 5 known, physically verified transdifferentiation experiments. And by analyzing pure mathematical vector shifts, ChromBERT successfully identified the exact perturbed key driver regulator in 3 out of 5 of those experiments.
Wait, really? Just from the math?
Completely, without the need for new physical ChIP-seq tests. It inferred the biological driver of cellular alchemy purely through geometry in a high-dimensional space.
That is mind-blowing. Okay, pulling this final section together for our listeners. ChromBERT isn't just an academic exercise. It is actively outperforming specialized models in isolating the exact genetic variants that cause complex diseases. And it is tracking mathematical embedding shifts to predict exactly which proteins you need to push to transform one cell type into another, doing it entirely in silicon. We're essentially watching virtual biology unfold. Math is identifying the specific proteins that turn connective tissue into immune cells.
It really is a massive leap forward.
But, before we declare the end of physical biology, we need to inject a dose of scientific reality here. What are the actual limitations of this model?
Yeah, it is crucial to ground this. ChromBERT is a monumental architectural achievement, but its ceiling is dictated by the data it learns from. As we mentioned earlier, it currently only models about 1,000 regulators because that represents the absolute limit of the public ChIP-seq data we have available.
Right.
There are countless other proteins, cofactors, and epigenetic mechanisms that are currently just invisible to the model.
Basically, if the paparazzi literally never took a single photo of an actor, the AI doesn't even know they're in the movie.
Exactly. Furthermore, because the researchers had to pool data across over 100 diverse cell types during pre-training to achieve sufficient volume for the transformer, the model inevitably smooths over and blurs some of the highly specific, nuanced behaviors unique to individual cell types.
So it's the inherent trade-off between massive generalization and hyperspecificity.
Right. And lastly, ChromBERT currently processes the genome in a linear one-dimensional format, those 1 kilobase intervals. But inside the nucleus, DNA is folded, looped, and compressed into incredibly complex three-dimensional architectures. Aligning 1D protein binding tracks with the functional realities of 3D spatial DNA organization, well, that remains a massive hurdle for future iterations of the model.
So what does this all mean? If we synthesize this entire journey for you today, we started with a massive void in biological data. We had the static DNA script, but the dynamic operations, the paparazzi photos of the protein actors, were fundamentally missing for most of human biology. And we've explored an AI that learns from 15% masked gaps to master the unwritten grammar of proteins. It utilizes complex mathematical concatenations as prompts to adapt to highly specific cellular stages. It maps causal disease variants better than DNA-only models, and it tracks high-dimensional vector shifts to predict the biological keys of cellular transformation.
It is a profound demonstration of how transformer-based foundation models can finally bridge the chasm between static genetic code and dynamic predictive biological function.
And I want to leave you, our listener, with a final provocative thought to mull over. We spent a lot of time discussing how ChromBERT predicts these missing pieces. But deep inside its transformer architecture, the model operates by calculating attention weights.
Right.
It continuously learns and maps exactly which proteins are paying the most attention to which other proteins across the entire genome. So theoretically, analyzing these attention weights could eventually allow us to map out the complete hierarchical command structure of a human cell from scratch, identifying the generals, the lieutenants, and the foot soldiers of human biology.
It's entirely possible.
So here is the question. If foundation models can eventually simulate the entire regulatory network of a cell perfectly in silicon, knowing every actor's motivation and every director's exact move, will we ever need to run a physical Petri dish experiment again? Or will the entire future of biological discovery happen strictly inside a hard drive?
That really is the ultimate question facing computational biology today. The very definition of what constitutes a lab is being rewritten.
It really is. Thank you for joining us on this deep dive. Keep questioning, keep exploring, and we'll see you next time.